Hello, this is Mr. Buss, and I'm going to continue instructing chi-square analysis. It's a statistical analysis that we use in AP Biology. Of course, you could use it in other sciences and other studies. And so in this one, we're going to look at M&M color variety. And just to save time, I already just took a bunch of M&Ms from a bag of M&Ms and dumped them out and just like sorted them into color piles. And it looks like there's 6 different colors, and each of those colors has a number, right? So 30 blue, 20 orange, 16 green, 11 brown, 33 yellow, and 57 red. And so now you could come about a null hypothesis in different ways. I came about the null hypothesis just by saying, without looking it up, I am going to say that I expect an equal color distribution of all colors. Well, you might not. So you might say, well, actually, no, M&Ms probably, they have certain like percentages for each color, and they do. And like you could look on their website maybe and find like what their percentages are. But I want to just come up with the assumption that without doing research ahead of time, just say my null hypothesis is that color varieties are equal. So like they make, equal batches of red, orange, blue, green, brown, yellow, and then you just dump them together. So I'm going to start with that. I'm going to see if that holds, So, it kind of looks like it doesn't hold. It kind of looks like there's a lot of reds and not a lot of browns, for instance. So we'll see if the chi-square value that we obtain is actually a high value, and see if we reject the null in favor of an alternative hypothesis. An alternative hypothesis could be that there is some non-equal color distribution of the M&Ms, okay? So we'll see where this takes us. So these are our observed values, okay? And so in class, we did group data, and I put on the board and stuff, and then we did a class data. But for this, I just dumped out some, okay? So what are our expected values? Well, our expected values are based on the null hypothesis and then the totals. And so we expect, based on the null hypothesis, we expect 1/6 of the total for each color, because that's what I said in the null. So first of all, we need to know the total. So I'm just going to quickly add these up here. So 30 plus 20, 30 plus 20 plus 16 plus 11 plus 33 plus 57 is 167 divided by 6. So I expect 27.8. Gosh, I kind of just want to round that. 27.8 of each color based on the null hypothesis. Okay, and I am going to round that. That's going to be annoying to work with. I'm just going to round that to 28. Okay, so those are our expected values, 28 of each color. So calculate the chi-square. So chi-square is observed minus expected squared divided by expected. And then we have this sum of all, which means that we have to do this calculation for each color variety. And there's 6 color varieties. So this is going to be done 6 times, and then you add them all up. So I'll try to write this quick so that the video doesn't take forever here. So expected is going to be 28 for all of them. And then, of course, I use my 30, 20, 16, 11, and 33 and 57 numbers for my observed. There we go. Now, get a number for all of those. So I'm just going to go through these quick. 30 minus 28 is 2. 2 squared is 4. 4 divided by 28 is 0.14. 20 minus 28 is 8. 8 squared is 64. 64 divided by 28 2.3. Oh, wait. Okay. 16 minus 28 is 12. 12 squared is 144. Divided by 28, 5.1. Eleven minus 28 is 17. 17 squared, 289 divided by 28, 10.3. Thirty-three minus 28 squared is 5. Squared is 25. Divided by 28, 0.9. 57 minus 28, 29 squared is 841. Divided by 28, 30. That was obviously the biggest one. That one's going to really skew this number here. So 30 plus 0.9 plus 10.3 plus 5.1 plus 2.3 plus 0.14 is 48.7. So that's our chi-square value. So chi-square is 48.7. So again, the larger the chi-square value, the less likely our observations are what we expect them to be. Excuse me. So using the 95% confidence interval of p 0.05, identify the critical value. So let me erase this from the penny. So 95% confidence interval, use the p-value of 0.05, it just tells us. The degrees of freedom is the number of possible outcomes minus 1. There were 6 color varieties. 6 minus 1 is 5. So we match up the columns and the rows, and we use 11.07 as our critical value. So 11.07 is the critical value, and that is less than our chi-square value of 48.7. And so because our chi-square value is really high, we reject the null hypothesis in favor of an alternative. And then you're like, well, what's the alternative? What could that be? Well, I wrote what an alternative could be. An alternative hypothesis is that there is some non-equal color distribution of colors because maybe research shows a different color preference makes us want to eat or buy more M&Ms. So there's probably a purpose or research behind the let's put more reds in, less browns, less greens, or whatever. And so I wouldn't be surprised if I went on M&M's website and I found, if I could find this information, I wouldn't be surprised if I found numbers favoring red. And I even, from what I've heard, think that there's 2 M&M factories perhaps that they maybe even use different color distributions, or there might be different color distributions in different bags. I know that they have holiday M&Ms and things like that, but I'm just talking about the regular ones. I'll just say briefly about this next one here. So part 3, let's apply this to genetics. So I mean, that's the whole point of chi-square for us, is that we want to use chi-square to interpret some biological observations. And so chi-square, I don't know exactly where you'll see it if you see it on the AP Bio exam, but it might show up in genetics, might even show up in mitosis, and I'll give you an example of that next week when we do a review. But with genetics, what you do is obviously you have your observations of offspring in like an F1 or F2 cross or something like that, or like a parent cross to get F1s or an F1 cross to get F2s. And then if you have observed values, right, which are here, then you can actually say, well, what did we expect? What do we observe? And do we observe what we expect, or is something going on here? Is our understanding of genetics of this concept off? okay? And so we can look at like a basic pea plant cross with complete dominant Mendelian traits, where we have like the trait of smooth or wrinkled seeds, right? And then I think we sometimes use round or wrinkled, but smooth is like also like another way to describe that. So if you take homozygous pure breeding or true breeding parents, that's like big T crossed with little t, right? And all of the, so that like you take the paintbrush, you get the pollen, you move the pollen back and forth, there's the cross, okay? So then all of the F1s are big T, right? Because that's what the Punnett square would show us. You know, we're gonna have, those crosses are gonna give us this heterozygous F1. But then if you take the F1s and cross them with each other, you get F2s. So that's a big T, big T cross, right? And when you do a big T, big T cross, of course, you get this ratio of like expecting 3/4 of them to be tall, right? And then like 1/4 of them to be short, because you only need one tall allele to be tall, right? So that's the F1 cross. Of course, the I'll just, you know, the parent cross was big T crossed with little t. That's why we got all heterozygous offspring, right? That was the first cross. That was the parent cross. Then you do your F1 cross, and then you get your F2. So your F2s, notice some of them are wrinkled, and here we got, you know, more of them are smooth. So we expect a 3-to-1 smooth to wrinkled ratio, you know, or 3/4 to 1/4 ratio of F2 seeds from our F1 plant. And so this is what Mendel actually recorded. It's kind of cool that we still have these results, that his actual results from like super long ago are still here. So then, you know, we're not looking at these ones. We're not looking at those. We're looking at these. Okay, those are observed. Those are observed results. Okay, and then our expected are from our ratios. So we do the same thing. You know, you say those are observed. Well, what was the total? Total was 5474 plus 1850. Observed is 7324. Well, I expect 3/4 of them to be spherical. So what's 3/4 of that? So that times 3 equals, divided by 4 equals, I expect 5, so this is the total. 3/4 of the total is 5493. I expect to be spherical. I can't write when I talk. And then 1/4 of the total, so 7324 divided by 4 is 1831. 1831 expected wrinkled in my F2s. And then I just say, okay, observed minus expected squared. So 5474 minus 5493 squared divided by expected, 5493, plus 1850 observed minus 1831 squared divided by 1831. Okay, do your math, get your answers, compare your chi-square value to the critical value, use a confidence interval of 0.05 of what it says in the problem, and see if Gregor Mendel's idea, which is right here, which is the null hypothesis, this is the null hypothesis, see if that's true, or see if something else is going on, so that you can apply chi-square to genetics, because you get observed offspring, and then you have expected ratios, and you can compare it to the critical value. And if the critical value is, so if you reject the null hypothesis, then what you're assuming is wrong, and it's probably like, in this case, we assumed Mendelian inheritance. Well, what if we got a really high chi-square value, which you're not gonna. You're gonna get a really low chi-square value. But what if you get a really high chi-square value here? You say, well, we reject the null. Maybe it's something else. Like maybe it's linked genes that we haven't learned about yet. Or maybe it's on the X chromosome, like a gender chromosome trait, like a sex-linked trait. Or maybe it's epistasis, right? So you can come up with some other ideas then if your chi-square value is really high and you reject the null hypothesis, if you reject the assumptions. So anyway, it can be a useful technique for confirming or validating whether or not results are close enough to what expected is. And of course, the eyeball test, like these numbers look really close. So I would expect that we would not reject the null hypothesis here.